So good afternoon everyone. Today we are starting with disorders of thyroid gland. And for that we are starting with first functional anatomy and physiology of thyroid gland, and we will also touch upon some points on investigations of thyroid disorders. So as you know, thyroid gland is an endocrine gland. We have learned about the differences of endocrine and exocrine glands in the previous class. Endocrine glands, the secretions, that is the hormones, are secreted directly into the bloodstream. And in exocrine glands, there is duct system. And through ducts, the secretions are secreted on the surface in exocrine glands like sweat glands and gastric glands. Okay. So thyroid is an endocrine gland, that means it secretes the hormone directly into the bloodstream, and it is responsible mainly for the maintenance of normal basal metabolic rate of the body. That means what is the function of thyroid gland is to maintain the BMR, that is the basal metabolic rate. So the main functions of thyroid is metabolism, growth and development, and intelligence. Also, it has a role in reproductive functions. So what is metabolism? Metabolism is the energy expenditure. It decides how much, at the base level, how much energy is required for various functions of the body. Okay. So to maintain the normal basal metabolic rate, the normal functioning of thyroid gland is essential. Okay. And anatomically, it is present in the neck. Everybody knows it. And it is of butterfly shape. Okay. It lies against the voice box, that is the larynx and the trachea. Trachea is your windpipe and larynx is your voice box. So, and just below the Adam's apple, the thyroid is located. Okay, the thyroid cartilage and cricoid cartilage lie just below the Adam's apple. Okay. And thyroid comprises of two lobes connected together in their lower two-third portion by a thin band of tissue known as the isthmus. The right and the left lobes are joined together by this thin band known as the isthmus, and sometimes on this isthmus a third lobe of thyroid is also present known as the pyramidal lobe, and which extends from thyroid gland up to the hyoid bone. Sometimes it is a rare exception, and variations, as you know, are also there. So usually there are two lobes, but sometimes the third lobe called pyramidal lobe is also present. Okay. And the uppermost part of the lobe extend to the thyroid cartilage. This upper portion is the thyroid cartilage and below is the cricoid cartilage. Okay. And the lowermost around the 4 to 6 tracheal ring. Isthmus is around first and second tracheal ring, and below that you can see fourth ring, fifth ring and the sixth ring. Okay. So it normally weighs around 25 gram in adults, and the normal variation can be up to 60 grams. That means from 20 to 60 grams it can weigh in adults, and each lobe being about 5 centimeters long. The length is 5 centimeters, breadth is 3 centimeters, as you can see in this diagram also, and 2 centimeters thickness, and isthmus is about 1.25 centimeters in height and width. Okay, so these are the measurements of the thyroid gland, and gland is usually larger in the women than in men, and it increases in size during pregnancy, because during pregnancy the energy needs of the body increases, so the gland also increases in the size. Okay, so here you can see the thyroid cartilage, and this is the cricoid cartilage. If you see from the levels of the vertebrae, that thyroid gland extends from T5 to T1. Okay, and in front from thyroid cartilage up to the fifth and sixth tracheal ring. Okay, and this is the pyramidal lobe. You can see in this diagram. This is the third lobe, pyramidal lobe. Okay, and these are right and left lobes. This is the isthmus which joins the two lobes. Okay. So it is also essential before going to the disorders to know the normal hypothalamic pituitary thyroid axis, or as in the short form HPT axis. Okay. So production of T3 and T4 in the thyroid is stimulated by thyroid stimulating hormone. Okay, the thyroid stimulating hormone which is secreted from the anterior pituitary, from the thyrotroph cells of the anterior pituitary in response to TRH, that is thyrotropin releasing hormone. Okay, so first thyrotropin releasing hormone is secreted. It is a tripeptide secreted from the hypothalamus, and it binds to the receptors on the anterior pituitary. Receptors are present on the thyrotroph cells, and it stimulates the thyrotroph cells to release TSH, that is thyroid stimulating hormone. These are all the trophic hormones. Trophic hormones means, trophic means to stimulate or to bring a change in the growth, nutrition, multiplication. Okay, so these are stimulating hormones. Thyroid stimulating hormone, as the name suggests, it will stimulate the thyroid gland. Okay, so it will bind on the receptors on the thyroid gland and will produce the thyroid hormones T3 and T4. Okay, also known as T4 is also known as thyroxine, and T3 is also known as triiodothyronine. Okay, why they are called T3 and T4? T3 because it contains three molecules of iodine, and in T4 it contains 4 iodide molecules. Okay, that is why it is called T3 and T4. Okay, and there is also a negative feedback of thyroid hormones on the pituitary. When the secretion of T3 and T4 increases, it gives a negative feedback to pituitary as well as hypothalamus, that there is adequate amounts of thyroid hormone in the blood, so you stop producing the TRH and TSH. So it will decrease the amounts of TSH. The TSH secretion is suppressed. Okay, and conversely, when the concentration of T3 and T4 are decreased, when the secretion is decreased, so they will send a message to pituitary and hypothalamus to increase the secretion of TRH and TSH. So the TSH level in the blood will be elevated. So there is a negative feedback of thyroid hormones on the pituitary. And what does this T4 and T3 does? The target tissues are heart, liver, bone, CNS, and all the nucleated cells of the body. Okay, and T4 is also converted to T3 in liver, muscles, heart, and kidney. Okay, so now you know the HPT, or the hypothalamic pituitary thyroid axis, that TRH is released from hypothalamus, which stimulates the thyrotropes in the anterior pituitary to release TSH, and this TSH will produce T4 and T3 from the thyroid gland, which will then act on the target tissues and glands. So when we go to the cut section of thyroid gland, histologically, the functional unit is round thyroid follicle, which is lined by follicular cells. You can see this round structure. These are thyroid follicles. The thyroid gland itself consists of lobules, okay, and these lobules contain the follicles. And the lining is by follicular cells. Inside is the colloid, okay, and these small white structures you can see are parafollicular cells, okay. So these lining the round follicle are follicular cells, and these scattered in between white egg-like cells are parafollicular cells. Okay. These follicular cells are also known as thyrocytes. They secrete the thyroid hormones T3 and T4. And these parafollicular cells, they secrete the calcitonin. Okay. Which is of no apparent physiological significance in humans. But they have a role in metastasis. In some types of thyroid cancer, the calcitonin levels in the blood increase and tell about the prognosis of the cancer. Okay. They also, calcitonin, as the name you can see, is also has a role in calcium homeostasis in the blood. Okay. So, three types of hormones are secreted from the thyroid gland. That is T3, triiodothyronine, T4, thyroxine, and calcitonin. So, three hormones are secreted, T3, T4, and calcitonin. Okay, these follicular epithelial cells, how they synthesize the hormones? They synthesize the thyroid hormones by incorporating iodine into the amino acid tyrosine on the surface of thyroglobulin, which is a protein secreted into the colloid of the follicle. So, these follicular cells require iodine. Iodine is a halogen and it is a trace element which the body cannot produce, so we require it in our daily diet. The iodized salts, the government has made plans about these programs to iodize the salts. So we have to take the iodized salt, and other food items also have iodine. So we have to take it in our diet. From the outside, it has to be taken, okay, because the body cannot produce itself. Okay, and this iodine will then get attached to the amino acid tyrosine on the surface of this protein known as thyroglobulin, which is produced by the follicular cells, and these are secreted in the colloid of the follicle. In this center colloid, the thyroglobulin will be released. Okay. And iodine is a key substrate for thyroid hormone synthesis. A dietary intake in excess of 100 mg per day is required to maintain thyroid function in adults. Okay, so it is a key substrate. It is very essential. Okay, so here you can see this is the structure of the thyroid follicle, the follicular epithelium. The center is the colloid, and in between these are parafollicular cells, T cells or calcitonin secreting cells, and in between are the capillaries containing the red blood cells. Okay, so the cut section through it, when we enlarge it, this hole is a follicular cell. Okay, this is colloid and this is extracellular fluid and this is the blood. Okay, so outside is the blood, this is extracellular fluid, follicular cell and colloid. So there are certain steps for the production of thyroid hormones. Okay, the first step is thyroglobulin, that is short form TG, is synthesized and secreted into the colloid of the follicle. So these follicular cells, you can see the number 1, TG is produced and secreted in the colloid. Okay. Then the inorganic iodide is actively transported into the follicular cell. That is the trapping of the iodine. This second number point you can see here at the lower boundary of the follicular cell, the iodide This inorganic iodide is trapped inside the follicular cell, where inside the cell it is organified. Okay, it is organified by certain enzymes. Iodide is transported to the colloidal surface by a transporter. So you can see the 3 number points here on the upper margin of the follicular cell. Okay, so this iodide is transported by a transporter known as pendrin, which is deficient in the pendrin syndrome, and it is organified by thyroid peroxidase enzyme. So two things are there here. One is transporter and another is thyroid peroxidase enzyme, which incorporates it into the amino acid tyrosine on the surface of Tg to form MIT and DIT. So here on the fourth step, you can see here this Tg on the surface of this is amino acid tyrosine. Okay, so amino acid tyrosine will get attached, incorporate the iodide to form monoiodotyrosine and diiodotyrosine. Okay. These iodinated tyrosines, these mono and diiodotyrosine will couple to form T3 and T4. When one MIT and one DIT will couple, they will form T3. And when two DIT will couple together, they will form T4. Okay. So these iodinated tyrosinosis on the surface of thyroglobulin Tg will form T3 and T4. Okay. Now see the fifth point. This whole structure gets endocytosed. This thyroglobulin on the surface of which is all the processes happening. This will get endocytosed, means it will get inside the cell again. It will get inside the follicular cell again. All this formation was happening outside the follicular cell in the colloid, in the follicular colloid. Now the thyroglobulin is back into the follicular cell. Okay. This thyroglobulin is now broken down or cleaved by proteolysis. That is the breakdown of proteins to free the iodinated tyrosine and thyroid hormones. Now this whole structure will break down to free the iodine and T3 and T4 hormones, and this iodide will be recycled by the follicular cells by the process of dehalogenation. Okay, the iodinated tyrosine is dehalogenated to recycle the iodide, and T3 and T4 are reduced. Okay, and this T4 can be converted to T3 by an enzyme known as 5-mono-deiodinase enzyme. Okay, so T4 can be converted to T3. And most of these T4 and T3 hormones are protein bound. That is, they are bound by thyroid binding globulin. Okay, TBG, thyroid binding globulin. Most of the hormone is attached to this TBG, and only less than 1% is free hormone, free T4 and free T3, and this small amount of less than 1% is the main hormone which exerts its pressure on the target tissue, like the increased metabolic rate by increasing the mitochondria in the nucleated cells, and it mimics like beta adrenergic action. For example, on the heart rate, it will increase the heart rate. Okay, tachycardia, that is also known as tachycardia and palpitations. It will increase the gut motility. Okay, and it will activate the central nervous system so that the sympathetic tone is increased. It will also cause bone demineralization. That means it will, the calcium is resorbed from the bone. The calcium is taken out of the bone. Okay, bone demineralization, and also it impacts the cellular differentiation. Okay, the growth and development and maturity of the cells also depends on this free T4 and T3. So this summarizes the functioning of the thyroid hormones. Okay, metabolism, growth, development, action on heart, action on GIT, and action on the CNS, as well as action on the bones. Okay, so for the normal functioning, these thyroid hormones T3 and T4 are required. So the thyroid secretes predominantly thyroxine, that is T4, and only a small amount of triiodothyroid. Okay, so the majority is T4 and only small T3. Approximately 85% of T3 in blood is produced from T4 by a family of monodeiodinase enzymes that are active in many tissues, including liver, muscle, heart, and kidney. So, T4 gets converted to T3 by 5 monodeiodinase enzymes, which is present where it is present? It is present in liver, muscle, heart, and kidney. So, the 5 monodeiodinase will come out of these liver, muscle, heart, and kidney and will convert T4 to T3. And selenium is an integral component of these monodeiodinase enzymes. Okay, so selenium is also required. So along with iodine, selenium is also required to convert this T4 to T3. Okay, T4 can be regarded as a pro-hormone. Okay, pro-hormone means before the production of the main hormone, pro-hormone is there. So since it has a longer half-life in the blood than T3. Because T4 gets converted to T3, that's why it is called as pro-hormone, and moreover it has a longer half-life than T3. Okay, approximately 1 week as compared to 18 hours of T3. Okay, so half-life of T4 is 1 week and half-life of T3 is 18 hours. Half-life means it breaks down at this much time. Okay, and then binds and activates thyroid hormone receptors less effectively than T3. Okay, so T3 is more potent than T4. Okay, and T4 is, it binds the thyroid hormone receptors less effectively than T3. And T4 can also be converted to the inactive metabolite, that is reverse T3. Okay, so one more, that is inactive metabolite is there, reverse T3. So T4 can also get converted to reverse T3. This T3 and T4, that is more than 99.9% circulate in the plasma bound to thyroxine binding globulin. That is TBG, thyroxine binding globulin. Okay, this is a protein. So most of the thyroid hormones are protein bound. And the minute fraction of unbound free hormones diffuses into the tissues and exerts its metabolic action. And the less than 0.1% free hormone that exerts its effect on the tissues and exerts its metabolic action. So now we will also see certain points on the investigations. Investigations, we will also deal with the specific diseases. When we deal with specific diseases, then, but in general, we are touching on some points. Okay. Some laboratories use assays that measure total T4 and T3 in the plasma, but it is increasingly common to measure free T4 and free T3. Okay, so as you have seen in the earlier slides that the main action is through free T4 and free T3. Okay, but some labs also measure total T4 and T3. Okay, the theoretical advantage of free hormone is that they are not influenced by changes in the concentration of the binding protein. Okay. So the main benefit of measuring only free T4 and T3 is that they do not get changed by the increase and decrease of the TBG. Okay, this TBG levels can be increased by estrogen, such as the oral contraceptive pills. If the woman is taking oral contraceptive pills, then TBG levels will increase, and this will show increased levels of total T4 and T3. But actually, free T4 and free T3 might be normal. So this is what is written. Okay. When the TBG levels are increased by estrogen, such as OCTs, this will result in raised total T3 and T4, although free thyroid hormone levels are normal. Okay. A circadian rhythm of TSH secretion can be demonstrated with peak at 1 hour and trough at 11 hours. So, like other hormones, it also has a diurnal variation. Circadian rhythm means it has a diurnal variation. That means levels change at morning and at night. Okay. So the peak of thyroid hormone production is at midnight, during the midnight. That is approximately from 11 p.m. to 5 p.m. And this median is given, that is the 1 p.m. in the midnight. It is at the peak and trough at the 11 a.m. Okay, from 10 a.m. to 5 p.m. there is the trough, that is the nadir. Okay, that means the low. Okay, but the variation is very small, so that the thyroid function can be assessed reliably from single blood sample taken at any time of the day. Okay, but this variation of peak and trough is very minute, so it doesn't impact the at which time the the blood sample is taken to measure the thyroid. So it is not necessary that for the thyroid function to be done, we should go at the fasting in the morning. Okay, so at any time the sample can be taken. Okay, and it does not usually require any dynamic stimulation or suppression test. What happens in the endocrinal disorders generally, if there is hormone deficiency, then dynamic stimulation tests are done. And if there is hormone increase or excess, then suppression tests are done. But for this, as there is not much difference in the diurnal variation, so any of such tests are not required, and the blood sample can be taken at any time of the day. Okay. Okay, and the relationship between TSH and T4 is classically described as inverse log linear. That means when TSH increases, okay, when there is increased T4, then TSH will decrease, and when T4 is high, then TSH will when T4 is high, TSH will decrease, and when T4 is low, then TSH will increase. Okay. And the anterior pituitary is, though very sensitive to minor changes in thyroid hormone levels within the reference range, and they are very sensitive to minor changes. Okay, like for example, in an individual whose free T4 level is usually 15 picomoles per liter, a rise or fall of 5 picomoles per liter would be associated on one hand with undetectable TSH and on the other hand with raised TSH. That means जो हमारी anterior pituitary होती है वो बहुत ही sensitive होती है to this hormone levels, thyroid hormone के जो levels होते हैं blood में उससे बहुत sensitive होती है. अगर किसी individual का normal जो है वो अगर 15 picomoles रहता है, तो अगर 5 picomoles उसका increase हो रहा है, ठीक है, T4 levels, free T4 levels अगर उसका increase हो रहा है तो क्या होगा, TSH will become undetectable. ओके, इतना ज्यादा जो एंटीरियर पिट्यूटरी सेंसिटिव होती है, अगर थोड़ा सा भी राइज हो रहा है तो वो TSH अनडिटेक्टेबल हो जाएगा, सप्रेस हो जाएगा. और अगर 5 मोल्स फॉल कर रहा है, अगर फॉल हो रहा है 5 पिकोमोल्स, तो वो क्या हो जाएगा, दैट विल कॉज रेज्ड TSH, उसमें TSH इंक्रीज हो जाएगा. फॉर दिस रीजन TSH is usually regarded as the most useful investigation of the thyroid function. इसकी वजह से जो TSH है वो most useful investigation है thyroid की. ठीक है. However, interpretation of TSH values without considering thyroid hormone levels may be misleading in patients with pituitary disease. ठीक है. TSH वैल्यूज को हमेशा हमें T3 और T4 के साथ में ही देखना है. अगर उसके बिना देखेंगे तो हम मिसलीड हो सकते हैं. अगर पिट्यूटरी डिजीज हो, पिट्यूटरी ट्यूमर हो या कुछ हो एंटीरियर पिट्यूटरी में, तो उस केस में हम मिसलीड हो सकते हैं. ठीक है. बिकॉज TSH इज इनअप्रोप्रिएटली लो और नॉर्मल इन सेकेंडरी हाइपोथायरॉयडिज्म. जब सेकेंडरी हाइपोथायरॉयडिज्म होता है, दैट मीन्स थायराइड के बाहर, सेकेंडरी मीन्स आउटसाइड द थायराइड ग्लैंड पैथोलॉजी हो, तो उस केस में TSH इनअप्रोप्रिएटली लो हो सकता है या नॉर्मल हो सकता है. ठीक है. और क्या होता है, मोर ओवर TSH में टेक सेवरल वीक्स टू कैच अप विद T4 एंड T3 लेवल. फॉर एग्जाम्पल, अगर लिवोथेरेक्सिन थेरेपी चल रही है, एटलीस्ट 2 वीक से चल रही है, तो TSH विल टेक अप्रोक्सीमेटली 4 to 6 वीक्स टू रीच अ स्टेडी स्टेट. ठीक है. और जो हेट्रोफिलिक एंटीबॉडीज होती है वो भी क्रॉस रिएक्ट कर जाती है होस्ट एंटीबॉडीज विद अफिनिटी टू एनिमल एंटीबॉडीज यूज्ड इन बायोलॉजिकल एसेस कैन आल्सो इंटरफेयर विद द TSH एसेज. ठीक है. वो स्पूरियसली हाई TSH शो कर सकती है. ठीक है. ठीक है. अदर मोडालिटीज कॉमनली एम्प्लॉयड इन इन्वेस्टिगेशन ऑफ थायराइड डिजीज इंक्लूड मेजरमेंट ऑफ एंटीबॉडीज अगेंस्ट द TSH रिसेप्टर्स. तो TSH रिसेप्टर्स प्रेजेंट होते हैं थायराइड ग्लैंड के ऊपर, उसके अगेंस्ट एंटीबॉडीज हो सकती है, उसको मेजर कर सकते हैं. जो ऑटो इम्यून डिसऑर्डर्स होते हैं तो उसमें ये TSH रिसेप्टर एंटीबॉडीज होती है, या फिर अदर थायराइड एंटीजेंस की होती है, थायराइड पेरोक्सीडेज की या थायरोग्लोबुलिन की एंटीबॉडीज होती है, उनको मेजर कर सकते हैं. रेडियो आइसोटोप इमेजिंग कर सकते हैं. फाइन नीडल एस्पिरेशन बायोप्सी कर सकते हैं इन केस ऑफ गॉइटर, एंड अल्ट्रासाउंड कर सकते हैं. ठीक है. तो ये सारे इन्वेस्टिगेशन हम यूजुअली करते हैं. ठीक है. तो अकॉर्डिंग टू द डिजीज किसमें कौन सी इन्वेस्टिगेशन लगेगी, किसमें एंटीबॉडीज मेजर करेंगे, किसमें नहीं करेंगे, वो वी विल टेक अप इन द नेक्स्ट क्लास. ओके. सो इन द नेक्स्ट क्लास वी विल टेक अप हाइपरथायरॉयडिज्म. ओके. सो हिबा, हैव यू टेकन द अटेंडेंस?
यस मैम.
ठीक है. सो इन द
मैम,
नेक्स्ट, यस.
मैम अगर किसी कंडीशन में TSH हाई हो और T3 T4 वैल्यू नॉर्मल हो तो उस कंडीशन में मैम क्या करेंगे?
हाँ, तो ये अगर TSH आपका
हाई हो
हाई हो, देखो यहाँ पे देखो
और
आप,
T3
ये
T4 वैल्यू नॉर्मल हो.
नीचे की जो नीचे से थर्ड जो है उसमें ये एलिवेटेड दिख रहा है और इसमें नॉर्मल नॉर्मल दिख रहा है. ठीक है. यही पूछ रहे हो ना आप? T3 एंड T4 आर नॉर्मल
TSH
एंड
एंड
TSH इज
TSH है.
एलिवेटेड, है ना? दैट मीन्स इट इज
यस मैम.
सब क्लिनिकल हाइपोथायरॉयडिज्म. Okay, we will take up in the next
ओके.
class. We will start with this and then we will move on to the hyperthyroidism. Okay, what patterns of thyroid functions you can get. Okay.
ओके मैम.
So when T4 and T3 changes, how the TSH level changes. We will take up in the next class. Okay. The query which you were asking was normal T3 and T4 with elevated TSH is present as subclinical hypothyroidism. Okay. The clinically it will not present. Clinical features will not show. Okay. Because there is normal values of T3 and T4, so that will exhibit the normal function. Okay.
यस मैम.
So hypothyroidism will
मैम
be
इस
there.
कंडीशन में मेडिकेशन इम्पोर्टेंट होंगी या फिर?
इसमें इम्पोर्टेंट नहीं होंगे क्योंकि आपके हार्मोन लेवल्स तो नॉर्मल है ना इसमें. अगर आपके रिड्यूस्ड हो,
यस.
अगर आपके रिड्यूस्ड होंगे हार्मोन लेवल्स तभी तो आप हार्मोन रिप्लेसमेंट करोगे ना उसमें. ठीक है. तो
मैम
कन्वेंशनली तो आप दोगे नहीं. अगर कोई सब
यस
क्लिनिकल
मैम.
है, दैट मीन्स जो टिपिकल क्लिनिकल फीचर्स होंगे हाइपोथायरॉयडिज्म के वो प्रेजेंट नहीं होंगे. ठीक है ना. हाइपोथायरॉयडिज्म मीन्स कि थायराइड थायराइड फंक्शन कम कर रहा है, अपने जितना करना चाहिए उससे कम फंक्शन कर रहा है ना.
यस मैम.
और उसके अगर क्लिनिकल फीचर्स ही नहीं है तो आप कैसे कह सकते हो. ठीक है. उसमें आप अपनी कॉन्स्टिट्यूशनल मेडिसिन या वो सब देख सकते हो अगर कुछ मिलता है आपको. तो वो सब दे सकते हो, या आप देख सकते हो कि उसका कॉज क्या है, कहीं सेकेंडरी कॉज तो नहीं है उसका, टीएसएच क्यों ज्यादा सिक्रीट हो रहा है, कहीं पिट्यूटरी में तो नहीं कोई प्रॉब्लम है, तो वहां का आप स्कैन करके देख सकते हो. So
ओके मैम.
we will take up in detail hyper and
यस.
hypo. Okay. Okay. Any other queries? अभी तक जितना बताया उतना समझ में आ गया आपको?
यस मैम.
ठीक है. तो आपके लर्निंग ऑब्जेक्टिव यहाँ से ये होने चाहिए कि आपको थायराइड की नॉर्मल पता लग गया है कि वो एक बटरफ्लाई शेप्ड ग्लैंड है आपका इंटीरियर नेक में यहाँ पे प्रेजेंट है. ठीक है ना. आपकी जो ट्रैकिया है उसके सामने प्रेजेंट है, बिल्कुल लैरिंग्स और ट्रैकिया के सामने प्रेजेंट है. दो लोब्स होते हैं, राइट एंड लेफ्ट, विच आर जॉइंट टुगेदर बाय स्टिमस. ठीक है. समटाइम्स एक थर्ड लोब होता है, पिरामिडल लोब होता है. ओके. और उसका नॉर्मल फंक्शन क्या है थायराइड का? उसका सीवीएस पे भी फंक्शन है, सीएनएस पे है. सबसे पहले उसका मेटाबॉलिज्म पे है, जो बेसल मेटाबॉलिक रेट है उसको मेंटेन करता है वो. ठीक है. एनर्जी एक्सपेंडिचर कितना करेगा, वही दैट इज द मेटाबॉलिज्म, कि कितना ब्रेक डाउन होना चाहिए, एनर्जी कितनी रिलीज करनी है. राइट. एंड सीवीएस पे मतलब इसका इम्पैक्ट होता है कि अगर ज्यादा हो रहा है थायराइड हार्मोन तो टैकिकार्डिया कर देगा, पैल्पिटेशन कर देगा. ठीक है. सीएनएस में इंटेलिजेंस में इसका इम्पैक्ट है. ठीक है. सिंपैथेटिक स्टिमुलेशन करेगा, सिंपैथेटिक नर्वस सिस्टम एक्टिवेट होगा. ठीक है. तो आप एक बार पढ़ लीजिए, इसको आप रिकैप कर लीजिए बुक में से. ठीक है. लेस देन 1 मिनट इज देयर. ओके. अटेंडेंस आप मुझे भेज देना ही हुआ. ठीक है. तो फिर हाउ थायराइड हार्मोन इज फॉर्म, वो हमने देखा कि फॉलिकल के अंदर कैसे बनता है. थायराइड फॉलिकल के अंदर, फॉलिकुलर सेल्स के अंदर ट्रैपिंग होती है आयोडीन की. ठीक है. थायरोग्लोबुलिन के ऊपर सरफेस पे अमीनो एसिड टायरोसिन होता है, जहाँ पे आयोडाइड बाउंड हो जाता है, और जिससे एमआईटी और डीआईटी बनते हैं, और वो कपल करके आपस में T3 एंड T4 बना लेते हैं. ठीक है. तो T3 एंड T4 या तो बाउंड होते हैं प्रोटीन से टीबीजी, या फिर फ्री होते हैं. और ये फ्री वाले जो होते हैं दैट विल एक्ट देयर